Next in the list of newer cell deaths, we have FERROPTOSIS. As the name says, it is made up of two words, ferrous and apoptosis. So the type of cell death is indeed apoptosis. That is, it is a programmed cell death. But the important thing that you need to know is that this programmed cell death, in fact, is not caspase dependent. It is instead iron dependent. So what is FERROPTOSIS? It is iron dependent programmed cell death. FERROPTOSIS is seen in cells in cases like stroke, neurodegenerative disease, and cancer. As for mechanism, so I'm going to give you a very simple overview of the mechanism of FERROPTOSIS because the detailed mechanism is well understood once you know free radical injury. So for now, a very superficial explanation. Within the cell, there is presence of ferrous iron. This ferrous iron goes through a specific reaction by the name of Fenton's reaction. See, it has Fe in the name, so it obviously has iron involved. Fenton's reaction. Due to Fenton's reaction, there is production of reactive oxygen species, also referred to as free radical. And these free radicals produced within the cell bring about free radical injury. But God has provided us with a master regulator. And the name of that master regulator is glutathione peroxidase 4. Glutathione peroxidase 4, it prevents the formation of free radicals so that free radical injury of the cell does not take place. But let's say, due to any pathological reason within the cell, like maybe in case of stroke or neurodegenerative disease or cancer, there is an immense rise in the level of ferrous iron. As a result, there is immense rise in the level of free radicals. As a result, the ferrous ions and the free radicals within the cell become so much so that they overwhelm glutathione peroxidase 4. They overwhelm the inhibitory action of glutathione peroxidase 4, which leads to free radical injury. Whenever there is free radical injury, there is disruption of the plasma membrane. As a result, the intracellular content, they leak out. And I've told it multiple times, when intracellular content they leak out, they cause inflammation. So here as well, morphological features are like that of necrosis. So FERROPTOSIS, in terms of morphological changes of the cell, are similar to that of necrosis, even though it is an apoptosis because there is a programmed cell death. Okay. Ultrastructure wise, two major changes take place in the mitochondria of the cell. Number one, there is loss of crista. So these infoldings within the inner membrane of the mitochondria are lost. Number two, there is disruption in outer mitochondrial membrane. As you can see here, there's disruption in the outer mitochondrial membrane. Okay. So free radical injury, a very important topic. I'll make a separate lecture on this. Next on the list of newer cell deaths, we have ANOIKIS. What is ANOIKIS? ANOIKIS translates to homelessness. And it makes complete sense because in ANOIKIS, the cell, when it is out of its natural habitat, that is the cell, when it is out of its home, it dies of apoptosis. For example, right here we have a normal bunch of cells. Out of these normal cells, this cell detaches. So there is detachment of that cell from extracellular matrix, or The cell is no longer in its natural habitat. As a result, it dies of apoptosis. So this is anoikis. Anoikis is the apoptosis of that cell which is no longer in its natural environment, in its home, in its natural habitat. In blue, I have shown cancer cells. Out of these cancer cells, this particular cancer cell detaches. However, a cancer cell is anoikis resistant. And that is why, even after detachment, it does not die of apoptosis. It instead travels and then shows metastasis. And that's the difference between normal cell and a cancer cell.